Good evening, everyone. It's 5:25 and we'll have everyone take their seats. And for the next 5 minutes, we'll play an introductory video of, and then we'll get the evening underway. So please take your seats and here's our video.
I just couldn't comprehend how one day I was fine and the next day I had been given 2 years to basically be left on this planet. I was teaching little ones at the time, 4.5 and 5-year-olds, so it was very busy. So for a while, I thought, I'm just tired. I haven't taught this age group for a long time. But it became obvious after quite a while that I was battling something, and you just know when your body is not good. I was racing off in the morning to school, and as I walked out the door, I took a step down. Unbeknown to me at this time, my right femur was very compromised and it took me off balance and I fell. And that was the beginning then of them taking a look internally. They said, well, look, let's do an ultrasound on your right leg. That's when they found a lesion. He took one look at the lesion on my bone and said, that's active. There's no way that's not doing something. So we're going to do a full body scan. From there, that's where they found the tumour on the lung, and they found out I had 4th stage lung cancer and it had metastasised to the bones. It was a big shock, massive. I'd never smoked, ever. And up until that point, life was pretty okay, normal. They straight away ascertained what kind of cancer it was, and I was very lucky because they had a targeted treatment. The treatment itself just gave me a huge moment of hope because it gave me time. It bought me time. It was a lifesaver. It just absolutely settled everything. The tumour started to shrink, everything seemed to dissipate, and life got back to some sort of normality. Very grateful for the amount of research that goes on. So here I am, nearly 11 years down the track, healthy and well and clear of any cancer. I do lots of creative stuff. I've got fantastic friendship group. I love life. The diversity in the types of cancer that go on require research to be pretty proactive because everybody's cancer is different. There's not one person's cancer that is exactly the same. So research is paramount in being able to move forward in looking at that diversity in the disease itself. I think when you've been taken to that point where you think you're going to lose it, it changes everything. It changes your whole perspective of how you're going to live your life. And I'm really good at it. I love it. I love my life. I really do.
We'll move on from the Prince Harry interview. So, ladies and gentlemen, it's my great honour to welcome everyone to this 3rd annual South Australian Immunogenomics Cancer Institute public lecture. My name is Professor Christopher Sweeney and I have the great privilege of being the director of the University of Adelaide's SAiGENCI. I'd like to give a warm welcome to everyone here today, our alumni, our donors, our partners, our SAiGENCI board members, university officials, our colleagues and collaborators at the University of Adelaide, University of South Australia, and the future Adelaide University, and our community research partners, as well as our hospital network Callan partnership as well. And I'd also like to welcome any participants who are online, and this has also been recorded. If any of your friends would like to listen to this later, just forward the link that you received. So thank you for coming for this evening. We've had better weather than we've had in the past, so this is great. Prior to commencing the evening's event, I'd like to share a few words of acknowledgement of country. We acknowledge and pay our respects to the Kaurna people, the traditional custodians whose ancestral lands we gather on. We acknowledge the deep feelings of attachment and relationship of the Kaurna people to country, and we respect and value their past, present, and ongoing connection to the land and their cultural beliefs. A couple of housekeeping items before we get going. Toilets are located within the Braggs building. This is the Braggs building. You can access the toilets by exiting the lecture theatre and turning left, and there are signs outside the building. So a bit of an update. The South Australian Immunogenomics Cancer Institute, SAiGENCI, is the newest cancer research institute in South Australia and the University of Adelaide's flagship cancer research program, a collaboration with Callan, the Central Adelaide Local Health Network, the Royal Adelaide Hospital and the Queen Elizabeth Hospital. And our goal is very clear. It is to have fewer patients develop cancer, so working on prevention and screening, as well as improve the lives of patients with cancer, to improve the therapies that patients have, less side effects and more effectiveness. As I look around this auditorium, it feels like just yesterday when we held the first lecture in 2023. Since then, we've recruited over 80 scientists from around the globe, ranging from postdoctorate scientists to full professors, as well as training 15 students to be the next generation of cancer researchers. SAiGENCI is underpinned by 4 research programs led by world-class leaders in their fields, and they are each coordinating groundbreaking and impactful laboratory and translational cancer research, which we will hand over to our clinical colleagues. So there's research going from the laboratory all the way to the clinic. And having made a substantial contribution to South Australia's overall pool of great scientists, SAiGENCI is now primed to compound this great progress with the merger with UniSA as part of Adelaide University. SAiGENCI is a new cancer research institute nurtured by the University of Adelaide and the Central Adelaide Local Health Network, focused on improving the care of patients with cancer or at risk of getting cancer. We are designing new programs focusing on the leading edge and having new, talented, fresh, energetic investigators to deliver on those goals.
There is a whole field of cancer epigenetics where the idea is, okay, we cannot do anything about that mutation because it already happened, but we can learn how to control the epigenome. If we know how to control that epigenetic layer, we should be able to reprogram any cell type into any other cell type. If you can convert a cell of the skin into a blood cell, why you will not be able to convert a cancer cell into a no-cancer cell?
When I started in prostate cancer research, there were very, very few treatments for men once they had progressed and become resistant to the standard treatment. In the last 10 years or so, all of a sudden, there's been a number of different agents that have been approved for prostate cancer, but unfortunately, it still does remain incurable. And so that's really what's driving us, is trying to understand, why does everyone become resistant?
Half of all cancers are linked to chronic inflammation. And so one of the visions that we have for our program is that we can develop new drugs that target the immune system and also suppress the cancer. And in doing so, we hope to improve the quality of life of millions of people, not only in Australia, but worldwide.
In the computational system oncology program, our hope is to be able to make something equivalent to weather forecasting model, but for cancer.
We want to find patients before they develop cancer and develop specific prevention or screening strategies.
One of the things that really excites me about SAiGENCI is the new interactions and collaborations that we can establish with neighbouring research institutes.
We have researchers from multiple disciplines working together in the same institute with one goal which is improve outcome for cancer patients.
We can all achieve much more in an environment like that than we could in isolation.
We're going to develop and deliver better strategies to improve patient outcomes, not only in South Australia, but all around the world.
And now to the main event. It is my great honour to introduce Professor Brendan Jenkins and Dr. Mara SAiGENCI from SAiGENCI's Tumour, Inflammation and Immunotherapy program. Both Brendan and Mara are two great examples of the incredible talent we have been able to bring to Adelaide. Professor Brendan Jenkins is head of the Tumour, Inflammation and Immunotherapy program, as well as head of the Cancer Immune Signalling Laboratory, CISL, at SAiGENCI at the University of Adelaide. He previously served as director of the Centre for Innate Immunity and Infectious Diseases at the Hudson Institute of Medical Research in Melbourne. Professor Jenkins' groundbreaking research has taken him around the world, speaking at conferences and publishing papers that have made an impact on how we understand cancer. He's one of the world's foremost experts in the role of the innate immune system in cancer development and treatment. Dr. Mara SAiGENCI is the group leader of the T-cell Immunotherapy Laboratory at SAiGENCI. After finishing her PhD at the University of Adelaide, she also worked at Monash University and at the Walter Eliza Hall Institute in Melbourne, focusing on modulating the tumour microenvironment to enhance anti-tumour immunity and improve response to immunotherapy. Dr. SAiGENCI's current research and her lab is focused on improving response to T-cell-based therapies and immunotherapies in solid cancers with a focus on lung and pancreatic cancer. And it's my great pleasure to have Professor Jenkins join the podium.
Thank you, Chris, for that very kind and generous introduction. Look, I also just want to thank and welcome everyone, both here tonight who are in person, also those of you online. It really is great to see, I guess, so many people who are interested in hearing our work at SAiGENCI about the inflammation and immunotherapy. So the vision of our program is really to understand how we'll get this working here. How the immune system drives various cancers, especially those that are associated with inflammation. And then this will obviously lead to the development of new therapies targeted against the immune system, which hopefully will aim to improve cancer patient outcomes and also the quality of life. So this is who we are. So tonight you'll hear from me talk about the innate immune system and inflammation and its role in cancer. And we'll also hear Mara talk about the role of immunotherapy in cancer with a particular focus on lung cancer. And you can see here all the various cancers that we work on in our program are amongst the most common and lethal cancers in the world. And we don't do this in isolation, of course. We do this in collaboration with the various other programs that are embedded within SAiGENCI. All right, so let's get to the heart of the matter, inflammation. It is the body's defense response to infection, injury, and also illness. So it can exist in an acute form, so short term, where you may have a sharp pain or tenderness in the body. You may have, say, a transient sensation of heat, temporary swelling in your limbs, or there may be flushing at the injury site. That's the acute form. Now, of course, there is also the chronic long-term inflammation, which is shown here. Now, this slide is not meant to convert people in the audience to being hypochondriacs. All I'm pointing out is that the symptoms of long-term chronic inflammation are many, and of course, there are various ones that we choose not to share with others. So what triggers inflammation? There are many triggers of inflammation. Probably the most common ones you'd be familiar with are infection, whether it be in response to microbes such as bacteria, viruses, and also fungi. Then you also have trauma, such as tissue wounds. Again, the easiest one to explain is a cut in the skin. And also you can have internal damage to cells in the organs inside you. And then we have various lifestyle factors such as excessive alcohol consumption. You've also got excessive exposure to tobacco or cigarette smoking. Poor diet, obesity, and also stress are also factors. And then we have other environmental factors such as sun exposure, which we're all aware of course, exposure to toxic chemicals and pollution, including in the air. So the thing to understand here is though acute inflammation, short-term inflammation is a good thing. Okay, we need it. It can last from days up to a week, and it's really critical to eliminate the infection, whether it be bacteria or viruses, and repair the tissue back to what it was normally. But of course, there are for some individuals and for reasons we still don't fully understand, whereas inflammation goes up and goes off very quickly, some people that inflammation just stays on for a long period of time. This chronic inflammation can last for months up to years, and this is when we run into problems with potential diseases, which I'll talk about shortly. Okay, so inflammation is a reaction of the immune system. Actually, before I go any further, on a show of hands here in the audience, please put your hand up if you know what or have heard of the innate immune system. Okay, a few of you, but also a lot don't. Okay, so hopefully at the end of the talk, if I ask the same question, there'll be a lot more hands will go up. So what is the innate immune system? It's essentially what we are born with. It's our body's first line of host defense. It's ingrained, it's hardwired into our genes, and it gives us lifelong protection from birth right to the end. And interestingly, it's actually highly conserved evolutionarily. So a lot of the components of the innate immune system in humans, they're actually shared with insects, plants, and even fungi. And these are just, so with the innate immune system, it's highly rapid. So it's a very quick response and it's non-specific. That means irrespective of the trigger, you kind of like get the same immune reaction occurring. And there are various components of the innate immune system. So you have these physical barriers. The most obvious one would be the skin. And you also have these mucous membranes which line our lung airways, and also our gastrointestinal tract. You have chemical barriers shown here. And then we have the first responders, which are cells within our blood system, some of which are shown here. And then we have these various molecules which are produced by these cells, and some of them are listed here, and you may be familiar with some of them, such as cytokines, which are one of the more common ones. And again, I think the best example of the innate immune system in action, again, is if we think about a cut to our skin, where bacteria will enter through the skin into the wound. And then what happens is your innate immune system within ours recognizes this, and it calls out an emergency response to these first responders. And they will then basically come into the wound, they'll kill the bacteria or the virus or whatever's in there, and then they'll eventually return the tissue back to normal. So that's the innate immune system. Now, the other arm of the immune system, so there are two arms of the immune system, is the adaptive immune system. Now, this is different. It's highly specific, it's slow, and it's memory-based. I'm not going to go into a lot of detail about this tonight because Mara, our next speaker, will talk about this in quite a lot of detail. But probably the easy example of the adaptive immune system is if you get a vaccination or immunization, say against a virus, whether it be flu or COVID, and your body mounts an immune response. That'd be the most simplest explanation. But as I said, Mara will talk about that in a lot more detail later on. So we have this situation that innate immunity can trigger inflammation. In an acute setting, it can be good. In a chronic setting, it can be bad. And inflammation is associated with a whole range of immune-related disorders, and up to, or at least 50% of all cancers, right? So over half of all cancers have an immune or inflammatory component to them. So here's a whole range of immune-related diseases, from autoimmune disorders, metabolic disorders, neurological diseases, bone and joint diseases, cardiovascular diseases, lung disorders, and other inflammatory and also infectious diseases. And you notice that quite a few of these have these red asterisks. Right? So these are the immune-related diseases or inflammatory diseases for which there is a known risk of developing cancer. So our research is really aimed at trying to identify new genes, in particular of the innate immune system, that drive the inflammation that promotes the cancer, okay? And in doing so, we hope to identify new targets for drug development, but also to try and discover biomarkers so that we can predict individuals who will develop these diseases and cancer before they actually physically develop them. This is one of the key goals. So my lab studies lung cancer, stomach cancer, and pancreatic cancer, and they're among the most common and lethal cancers in the world. And these cancers all have one thing in common, and that's the immune system, in particular chronic inflammation. So COPD, or chronic obstructive pulmonary disease, is a risk factor for lung cancer. Gastritis, as many of you are probably aware, is a risk factor for stomach cancer, and pancreatitis is a risk factor for pancreatic cancer. And I'll actually finish off shortly a story we've got in the lab about pancreatic cancer. So I've hopefully convinced you that inflammation is pretty important in terms of driving a lot of cancer. So this would lead us to ask the question, well, can anti-inflammatory drugs be used to stop the risk or reduce the risk of cancer? Now, the answer to that question is complicated, and it's yes and no. And I'll quickly talk you through this slide. So what we essentially have here is a summary of all the various types of anti-inflammatory drugs that have been studied in early stage through to late stage 4 clinical trials to investigate their impact on reducing cancer risk. So we have some targeted therapies here against cytokines. We have statins, which are cholesterol-reducing drugs. We have metformin, which is a drug that's used in type 2 diabetes to lower blood sugar levels. Non-steroidal anti-inflammatory drugs, NSAIDs. You'd be familiar, of course, with aspirin, which is one of those. Then we have these anti-infection agents, so antibiotics, which obviously are against bacteria. And then we have vaccines and antiviral drugs, which are against viruses. And then, of course, we do have natural supplements, which people use. So if we turn our attention to this fellow over here, and in particular, these red dots. So we've got liver cancer, gastric cancer, and cervical cancer. These are the cancers which seem to have had a pretty good effect in terms of these anti-inflammatory drugs lowering the risk, right, of developing these cancers. And an example of this also relates to the fact that this reduced risk seems to align strongly with the use of these anti-infection agents, okay, because these infections will drive inflammation. So we all know about hepatitis B vaccines are very good at protecting against liver cancer. We all know about human papillomavirus, how successful that has been in protecting cervical and other cancers. And then we have antibiotics, for example, against Helicobacter pylori, which is a bacteria that accounts for about 3/4 of all gastric cancer patients throughout the world. So there are certainly situations in some cancers where these anti-inflammatory agents can reduce the incidence of cancer and increase life expectancy. However, it's important to note that many of these anti-inflammatory agents do not have any impact on reducing cancer risk, and many of them have undesirable side effects, especially those that target inflammation broadly, because you're dampening down the immune system, which means you're also targeting parts of the immune system you actually need to maintain a healthy lifestyle. So there is still a clear clinical need to identify regulators of the innate immune system as anti-inflammatory agents to treat in cancers together with existing chemotherapy drugs. Okay, so we're not talking about single agent treatments here, it's together with existing treatments. Right, so I'm going to finish off on the last half of this talk about an active project we have in the lab related to pancreatic cancer. And if there's one word I want you to all remember tonight, in addition to innate immunity, it's this, inflammasome. And I'm pretty sure not many people have heard of this inflammasome before. So I want you to think of this as a multi-protein factory, okay, of the innate immune system. So these inflammasome complexes, they comprise 3 separate types of proteins in this complex. One's called a PRR, which is short for pattern recognition receptor. This ASC adapter, which is critical. And then we have this caspase 1, which is an enzyme. So what do we need and why do we have factories? Well, they comprise of components, machinery, and equipment to assemble an end product that then is distributed and delivered throughout the world. So knowing that, when a stimuli such as a microbial infection comes along in your body, this particular PRR will sense it, recognize it, it becomes activated, it then will recruit this ASC protein. ASC will then recruit caspase 1. So you now have this complex. And what happens is that complex then binds to another complex and it binds to another complex. And before you know it, you build up this massive complex, these huge aggregates, which are called inflammasome specks, because you can actually see them under a microscope. That's how big they are. And these inflammasomes are critical for the production of potent proteins in the body called IL-1 and IL-beta and IL-18. Think of these as those first responder molecules I spoke about earlier. And what happens is when they're made, they're then distributed throughout the body and delivered to various organs in the body where they have their effect, which can be potent in terms of driving inflammation and also cancer. So we know from work over the last more than a decade or so that increased activation of inflammasomes drives inflammation, which is linked to all of these inflammatory related diseases. What about cancer? It's not as well known, the role of inflammasomes in cancer. And so we've been working on a story on pancreatic cancer to investigate the role of these inflammasomes. And so what we can do is from patients with pancreatic cancer with their tumours, we can extract DNA. DNA, of course, contains all the genes that are present in that tumour. DNA is normally transcribed to RNA. RNA is translated to protein. And using various techniques in the lab, we can then measure the amount of ASC at the RNA and protein level, and also IL-18, remembering these are key components of this inflammasome complex. And indeed, what we found was that in these pancreatic cancer patients, they have increased levels of ASC at all stages of disease compared to these normals, which is normal pancreas from individuals which don't have cancer. And if you focus on this red line in particular, what this shows is that patients who have high levels of ASC have low survival, okay? High ASC, low survival. So having ASC at high levels is a bad thing in pancreatic cancer, and we believe it's a prognostic marker for poor survival. Now, using other techniques in the lab called immunoblotting and also immunohistochemistry, we can assess the levels of ASC at the protein level in the tumours from these patients. You can see there's high levels of ASC by immunoblotting and by immunohistochemistry compared to normals, which again is normal pancreas from patients or individuals who don't have cancer. And this brown staining here actually indicates ASC expression, or the amount of ASC protein that's made in individual cells. So you can see there's a lot of it in the tumour compared to the normal. And using another technique, immunofluorescence, we can identify these ASC specks, which are these yellow dots you can see here. So these big aggregates I was telling you about before. So they're elevated in the tumours of patients. And using another assay called ELISA, we can detect increased levels of IL-18 in the serum of these patients. So clearly we have evidence that these inflammasomes are upregulated, okay, in pancreatic cancer. So we need to prove this further in the lab. So what do? We turn to our preclinical models. So this is what a normal pancreas would look like with no cancer under a microscope. This is what the pancreas looks like in our KPC pancreatic cancer model. It's essentially a tumour, massive tumour, okay? And in this tumour, we see high levels of ASC by immunofluorescence and high levels of ASC by immunoblotting. But importantly, if we use genetic engineering, okay, in the lab, and we delete the ASC gene, okay, so there's no more ASC in this KPC pancreatic cancer model. And you can see that here, the protein's been completely eliminated. This pancreas now almost looks like a wild type pancreas. So we've dramatically reduced the formation of tumours in this model just by deleting one gene out of over 25,000 genes, one gene of the immune system. And what we show here is that using an antibody that detects immune and inflammatory cells, all this brown staining indicates inflammatory cells that are in the tumours of this KPC model, whereas in the absence of ASC, again, we don't see any of these tumours. So ASC is driving inflammation, it's driving tumorigenesis in the pancreas. Now, that's all well and good genetic engineering, but we obviously need a drug to try and target ASC, because that's what you want to do if you want to move this into the clinic. And we were very fortunate to obtain a drug that's called a nanobody. Now, for those of you who don't know what nanobodies are, we have to turn to our camelid friends here, the camel, the llama, and the alpaca. And in addition to having conventional large antibodies that we're all familiar with, these guys make these tiny little antibodies called nanobodies. And these nanobodies have a lot of advantages over conventional antibodies. And fortunately, through a collaboration with Florian Schmidt in Germany, we have access to this nanobody. And yes, Florian is the one on the left with the less hair, for those of you who may not know. And what we've been able to show with Florian in particular through his work is that he's shown that if you actually treat models in the lab for gout and rheumatoid arthritis, which are inflammatory diseases, these nanobodies actually protect these models so that you don't develop these inflammatory diseases. And this is very complicated. You don't need to know this. All it shows is that these nanobodies work by targeting these ASC inflammasomes both outside of the cell, this is a cell here, and also inside the cell. But the big question is, which remains unknown, can these ASC nanobodies be used in cancer? Because no one knows this. So we devised this experiment with Florian where in our model, Our pancreatic cancer model, we treated for 9 weeks with this nanobody and vehicle control. Remember, the nanobody will essentially dissolve away these aspects. And what we found was that in contrast to the control model, which received this vehicle saline, you can see that with the nanobody, the pancreas now is more or less normal. So the nanobody did in fact dramatically reduce the formation of these tumours. And this is coincident with a large reduction in the amount of vascular inflammasomes forming here. You can see with the nanobody. And importantly, we also see reduced numbers of inflammation. So now we have access to a drug that we can use and develop further to try and target this even more in the lab. And that brings me to this slide. So I've only got a couple of slides left. I thought I'd introduce this slide because this is what we call the bench to bedside pipeline, okay? It's basically the timeline by which you discover a gene in the lab that you think may be involved in a disease. And then you identify a drug that could target that gene or that protein. And then you take it all the way through to the preclinical work in the lab till you get to the point where hopefully you can advance it into the clinic. Now, I've just given some arbitrary numbers here as a guide. This is not definitive. It can be longer, it can be shorter, but generally you're looking at about a 10-year plan at least to get to this point. And with this work at the moment, with these nanobodies and the work we're doing in pancreatic cancer, we're about this point. So we've still got a fair way to go, but the evidence is looking pretty promising at this stage, the clinical work and the preclinical work. And also just wanted to highlight that in addition to pancreatic cancer, we have evidence we published previously that inflammasomes may be important in driving gastric cancer. So this could be applicable to other upper GI cancers. And that just leads me to finish up on this slide. So I'm now going to ask you, who here in the audience knows what innate immunity is? There we go. A few more people. Excellent. So now we've all learned something tonight. That's great. So we know that inflammation triggered by innate immunity can be a good thing when it's in an acute setting. We need this to fight infection and heal tissue. But we also know that increased activation of inflammasomes can trigger chronic inflammation, which can lead to pancreatic and most likely also stomach cancer. But now we have access to this nanobody drug, which hopefully will allow us now to develop this further to reduce the risk of these cancers in collaboration with our friends in Germany. And finally, that just leaves me to thank the funders who we rely on, obviously, for this work to continue and a lot of the other work in the lab. But most importantly, it is these people here in the lab, a few of them, they're the real stars, they're the ones who do all the work. So I'm always indebted to the guys in the lab because without them, none of this would be possible. I'll leave it there and thank you for your attention. And now it is a great pleasure for me to introduce Tumour SAiGENCI, who will be talking about lung cancer and immunotherapy. So if we can please welcome Mara to the stage. Thank you.
Thanks, Brendan. Hi, everyone. I'd just like to echo what Chris and Brendan have said before and welcome you all here tonight. Thank you for joining us. So my name is Dr. Tumour SAiGENCI. I'm a group leader within the Tumour Inflammation and Immunotherapy program at SAiGENCI. And today I'm going to talk to you about how we can harness the adaptive immune system to fight cancer and in particular lung cancers. So just very briefly before we get into the lecture, I thought I'd just tell you a little bit about what my research journey has been to date. So I did my PhD over here in Adelaide and finished in 2020. And this PhD was really focused on the blood cancer multiple myeloma. And multiple myeloma, it's in the name. You can see here by this PET scan for this patient, these patients have multiple tumours all throughout their skeleton at diagnosis. And so really a key question for us here was, how is this the case and how are these cancer cells spreading so rapidly throughout the bone? So what we focused on this receptor called CCR1 and showed that if we remove this receptor, we can completely prevent these cells from moving from bone to bone. And importantly, we found a drug that we can use to prevent this process and slow disease progression in patients. In 2020, right at the beginning of the first of, I don't even know how many lockdowns, I decided to move to Melbourne, which was work-wise good, personal life not so much. But this was to work at Monash University looking at breast cancer. And in particular, this project is looking at obesity because breast cancer is one of many cancers that's highly driven by obesity. And what we were looking at here was what was happening in the immune system when patients were obese and had cancer. And what we found is that a lot of these tumors actually were quite immune cold. And so there was not a lot of immune cells when a patient was obese. And so we were looking at this protein called PDPN2 and found that if we remove this protein, we can actually turn these tumors immune hot. So we get a lot more immune cells into the tumors, and this improves response to immunotherapy. And these, since I finished this research, there's been PDPN2 inhibitors that have been developed in the US that are now being used for multiple different cancers. And then I moved, still in Melbourne, but to the Walter and Eliza Hall Institute. And here is where it's really been the foundation for the new work I'll be doing here in Adelaide, looking at lung cancers. And in particular here, we're using these functional screens to try and find new drug targets that can improve immunotherapy for lung cancers. And so, yes, as I said, this is some of the data I'll show you today, and it's really the basis of what we're looking at here in Adelaide. So if we think about the innate and the adaptive immune system, Brendan's given you a really good overview of how the innate immune system works, but it works in concert with the adaptive immune system. And really, the adaptive immune system is actually activated by the innate immune system when it's no longer able to control an infection. So it recruits two main cells, the B cells and the T cells. So the B cells produce antibodies that neutralize whatever pathogen, so whether that's a virus or a bacteria in our body. But then importantly, and the focus of this talk are the T cells, which are cells that recognize, that get activated by the innate immune system to recognize the pathogen and can directly kill infected cells. And so really we've got this sort of powerhouse that's capable of clearing pathogens. So can we harness this power to fight and even clear cancer from our system instead? So to start to think about this, we need to look more closely at our T cells. So we've got different types of T cells in our body, but the main focus for immunotherapy are these cytotoxic T cells. So you can think of them as the cells that are able to produce toxic agents and kill their direct targets. So this includes, like I mentioned, cells that have been infected by a virus or bacteria, but it also means they have the capacity to kill cancer cells. So you can think of them as these killer T cells that we can harness to directly kill cancer cells in our body. And importantly, as I mentioned before, these T cells aren't always active. That would be a problem that then leads to things like autoimmunity and high levels of inflammation, like Brendan was discussing. So we don't want these overactive T cells. We want very specifically activated T cells. And so this majority of the time happens through the innate immune system, where cells called dendritic cells actually show the T cells what they need to attack, and this activates them. So what does this look like in action? So this is sort of a stepwise of what happens when we get exposed to a bacteria or a virus and had the response of the T cells. So I'm going to show you what happens in this sort of context, and then how that's different when we talk about cancer. So if we start from the start over here, we've got a virus that sort of enters our body, and our dendritic cells that recognize this virus. They then become activated and take these viral particles to the lymph nodes to show it to the T cells, and this leads to their activation. So when we have active T cells, they then enter the bloodstream and go back to the site of infection and directly kill our infected cells and help clear the infection. The B cells work in concert with this and also migrate to the site and produce the antibodies to neutralize the infection as well. So what happens in cancer? So it's similar, but of course not the same. Our cancer cells, just like a virus, actually produce these molecules that the dendritic cells do recognize. So they're able to recognize that the cancer cells are not normal and that something is wrong. And when they take these particles, they can show it to the T cells and activate them. So the T cells can now migrate back to where the cancer is and attack the cancer cells. Of course, this is best case scenario, not really what actually happens in practice. So we look a lot more closely at what's happening in this context. Our cancer cells are very clever. And really, they are obviously our own cells. So our body has systems already in place that actually stop us from attacking our own cells. So to then try and get the immune system to attack cancer cells is actually not an easy thing to do. And one of the things that tumor cells do to avoid being attacked by the immune system is they are able to have these inhibitory signals. And the way these work is that essentially the cancer cell can express an inhibitory signal on its cell surface. One of the most common ones of these, or most popular, I suppose, for immunotherapy is PD-L1. And this inhibitory ligand binds to the PD-1 receptor on the T cell and shuts down any immune attack. So this is where immunotherapy comes in. So immunotherapy, or one form of it, is actually the delivery of antibodies that block this inhibitory signal. And so they can either block the PD-L1 on the tumor cell or the PD-1 on the T cell. And this actually allows the T cell to mount an immune attack now. So it recognizes the cancer and it can now attack because there's no more inhibitory signal. So you can think of this type of immunotherapy as a way of boosting the activity of our T cells that they already innately have. So when we think about what we see in the clinic with immunotherapy, the sort of poster child for solid cancer is melanoma. So with melanoma, patients that are treated with immunotherapy to get really long-term response rates and the disease is controlled. But unfortunately, that's not the case for the majority of other solid cancers. Only about 25% of people with solid cancers treated with immunotherapy actually respond to the treatment. And when they do respond, these response rates are often quite short-term, and you're not getting that sort of long-term durable response to treatment that we want that increases patient survival and gives good quality of life. And one of these cancers that has very little positive benefit from immunotherapy is lung cancer. So lung cancer is one of the deadliest cancers. Only about 15 to 20% of people are still alive 5 years after diagnosis. And it is the 5th most commonly diagnosed cancer in Australia and around the world, but it actually accounts for the most cancer-related deaths. So it's a really horrible disease. And I know what you're all thinking. Yes, the majority of lung cancers are associated with smoking, but really what we're seeing now is actually an increase in the incidence of non-smoking or non-cigarette smoking lung cancers. So already we see that 1 in every 3 women and 1 in every 10 men diagnosed with lung cancer actually have no history of smoking. And Glenda's story is an example of that. But what we also is a bit scary now is that we see this high incidence and prevalence, of vaping that's really increasing in our society. And still right now, we have no idea how that's going to affect the incidence of lung cancer, although we have a fairly good idea that it's definitely going to be a huge issue. So lung cancer still really remains one of these critical cancers to investigate to see if we can find new therapeutic options given its increasing incidence. So if we think about lung cancer, well, can we use immunotherapy? Can we harness immunotherapy? Can we improve it for lung cancer? So we need to answer these questions, we need to look back more at the biology and see, well, how do lung cancers actually evade the immune system? So they can do this in 2 different ways. One of them is that they have the ability to hide. So I showed you earlier that cancer cells can express PD-L1 as an inhibitory signal to prevent the T cells from attacking them. We can actually kind of think about it as literally an invisibility cloak. So when a tumor cell is expressing this ligand, the T cell really cannot recognize it at all. And the other thing about these cancers is that they're what we call immune deserts. So there is almost no immune cells present or surrounding the cancer. And what this means is that even if the T cell is capable of mounting an anti-tumor response and is responsive to immunotherapy, there's actually not enough of them to see any effect on the tumor or to control the disease. So how can we overcome these 2 barriers? So we have our lung cancer that is immune cold and hiding from the immune system. So what can we do in the lab to try and figure out how we can improve the situation to get these cancers to respond to immunotherapy? So one of the things we can do is called functional screening. And so this is when we try to find new drug targets that can either increase immune infiltration or unmask the cells, or ideally do both. And so the idea here is we'd be turning our sort of immune deserts into these lush oases that's going to respond to immunotherapy. So to be able to conduct these functional screenings, we need to model this in the lab. So we need to be able to grow our T cells and our tumor cells together. So if we go back to our killer T cells, this you can see is a picture here of red cancer cells. They're marked in red, and our T cells here in green. And this is where we've just put them together in a dish. But over that 72-hour period, now you can see over here, there's no more red signal. So we've got no more live tumor cells. And our T cells have very specifically targeted and killed all our cancer cells. So using these types of co-culture models, we can grow both our T cells and our tumor cells together and start to really untangle their behavior and what allows them to kill the cancer cells and what doesn't. So to do these functional screens, I'll talk you through this. This is a sort of overview pipeline of what these functional screens look like. So if we start over here, we've got our immune cold lung cancer cells, and we use what are called molecular scissors. So this is where we can target specifically one gene in each cell and do this for every single gene that we have in our body. So this is what they look like. So the molecular scissors, so we've got our Cas9, which is an enzyme that is guided to a particular target gene by what's called a guide sequence, which is a complementary bit of DNA that matches the target gene. So by this way, we've got our Cas9 that gets directly taken to its target gene, and here is where it cuts the DNA and renders that gene inactive. And so if we repeat this process, but for every single gene, so there's about 20,000 genes in our genome, if we repeat this 20,000 times, we now have a pool of cells in the lab, each one with one loss of one gene. And so now what we can do is if we apply T cells on top of these cells, we can then look for what happens and what genes increase T cell killing and what genes increase resistance to T cell killing. And of course, the ones that we want are these ones that increase T cell killing. So you can think about it as if that gene is gone, and that T cell is capable of killing that cancer cell now, then if we use a drug against that gene, we're going to ideally improve the T cell's ability to kill the cancer. So these are sort of new drug targets that we're interested in. So this is some data that we've generated in the lab where you can see how this sort of works. So here we have the log. So from 0, so any gene that's up here is completely unaffected by T cell killing, all the way down to these sort of 6-fold reduction, where here is our top hit, where we see that when we apply T cells, we get a 6-fold reduction in the expression of that gene. So this is a gene that, when lost, increases T cell killing. And it's called a dub, and these dubs are involved in the cell's ability to recycle proteins. So just by saying that, you can already think, okay, it actually affects a lot of different signaling pathways and really just remodels the entire cell when it's lost. So the hypothesis here being that it's remodeling the cell in a way that is helping the immune system now recognize it. So we wanted to confirm that, yes, when we remove this dub, that we do get an increase in T cell killing. So we went back to our original T cell co-culture assays, where we have our immune cells together with our our cancer cells together with our T cells. But this time we've got our tumor cells that are controls that have no changes, and our tumor cells that have the dub deleted. We then apply T cells onto these tumor cells at different ratios. So all the way from 8 T cells to every 1 tumor cell, so lots of T cells, down to 1 T cell for every 1 tumor cell. And what we're looking for here is sort of a dose-dependent killing effect, looking for whether as we add more T cells, we get more killing when we have this dub deleted. And we can read this out and really basically looking at something like a crystal violet stain, where when we get full staining and purple dish, this means that all our cancer cells are still alive. And you can see that we can see when we've got only 50% of them alive, and when we've got no staining at all, we've got complete death of our cancer cells by the T cells. And so this is what it looks like for our control cells. So already you can see there's a lot of purple. These cells are really resistant to T cell killing. Even at this ratio, we've got 8 T cells for every 1 tumor cell. still there's about 30-something% of tumor cells that are still alive. But when we delete this dub, now we're suddenly seeing almost complete killing of our tumor cells. Even at these really low T cell ratios, where there's barely any T cells there, this is still able to really recognize and directly kill these cancer cells now. So that was really exciting. So we wanted to know, okay, well, how is this happening? What effect is there on the T cells when they're exposed to these tumor cells that don't have the dub anymore? And so to do this, we turned to our preclinical models in the lab and took tumors from control versus dub deleted and looked at the function of the T cells. And what we found is that there's now actually a significant increase in the number of T cells, here in purple, that have come into the tumors when the dub's been deleted. And you can see that nicely here in these pictures where this is our T cell population and in the controls, and it's essentially doubled when we have deletion of this dub. And in terms of the activity of the T cells, when a T cell recognizes its target and directly kills it, it produces these cytokines called TNF and interferon. And so we can measure the levels of those in the tumors, and we can see that now we have in these purple bars really high production of these cytokines, so the T cells are really wiping out this tumor. So what effect does this have on immunotherapy? So we've got more T cells, we've got more activated T cells, everything that we want when we're thinking about getting a response to immunotherapy. So we wanted to have a look at what happens to tumor growth over time. So this is size of the tumors versus time. So here you can see in black that control tumors really grow really rapidly and exponentially quite quickly. And when we treat control tumors with immunotherapy here in this dashed black bar, we do get a reduction and slowing in the growth of these cancers. But what was really dramatic in these purple bars is that with deletion of the dub alone, or with the deletion of the dub with immunotherapy, we get a four times decrease in the size of these tumors. And this also translates to survival. So when we have just control to immunotherapy treated, we have a 13-day survival increase. But when we delete the stub, we're a doubling of that and closer to 28 days. So this just shows that we can find new targets for immunotherapy, and we can improve the responses to immunotherapy by targeting these immune modulatory genes. So this is just one example of how we can use the power of these functional screens to find new targets for immunotherapy. Because at the moment, we're sort of sitting in this scenario where patients treated with immunotherapy, and you're not really getting any response to treatment or very minor response to treatment. But using our tools that we have in the lab, where we can use these really functional screens where we directly look at the activity of the T cells, how we can get them to kill our cancer cells more, and the drugs that we find, or the genes that we find in these functional screens, we can then one day follow Brendan's pipeline that you showed you, generate a drug against these genes that can be given to a patient in combination with immunotherapy to now actually get these activated T cells that are able to clear the cancer and you see disease regression. So with that, I'd just like to thank the lab over at WeHi in Melbourne, the Southern Lab, where a lot of this work has been done. Of course, our funding bodies and the new lab here in Adelaide that's only just started up. So yeah, it's all exciting times to come. So thank you very much.
So we have time for question and answers, and no one gets a graduation ceremony from all the science you've just heard with the graduations happening outside, but that was amazing science, and come back for more in future years. So we've got roving microphones, and I'll stand over here so I can see people. And so who's going to kick off first with their question? We have one down here on the left, Gia. Gia's got her sand shoes on, runners, and coming at speed.
There's another one there, Chris. If you want to use
Yeah, I was just thinking that too, but Gia's gone a bit faster, I think. And Anne, there's a lady on the right here, Anne. So first, someone put your hand up on the right so Anne can come to you, actually. So please.
I'm just wondering about the issue of delivery of the immunotherapy. Overseas, they're looking at direct intratumoral delivery. to bypass the problems with perfusion in the tumour. Is anyone doing any research looking at how the immunotherapy is being delivered? Either person?
Yeah. You probably talked to what Yannen's doing, right? Yeah.
One of the groups oh, no, who's that? Giving feedback. Actually, I was told I have to stay away from Mara because our microphones may talk to each other and give feedback. Probably have to stay away from you too, Chris. One of the groups in our program, Yan and Yang, he is actually working on doing that kind of thing, localized delivery. But the main thing with that, I think that it's difficult because if you're talking about doing that with tumours that are inside the body, it's very hard to do intratumoral delivery. I think it's more, correct me if I'm wrong here, it's pretty easier, Chris, if you're going to do it in skin cancers or where the tumours can be more accessible to that kind of intratumoral delivery. I'm not overly familiar with what people are doing internationally in that space related to your question. I don't know if you have any thoughts on that, Chris, as a clinician.
So as a medical oncologist who does do drug development, I'm a little bit closer to this, so I'll try and answer it. Actually, right here in our own little old town of Adelaide and throughout Australia, probably the most advanced way is nanoparticles being developed that can encapsulate proteins or various therapies and be transported around the body through the blood system. And then those nanoparticles tend to get taken up into the vasculature, the blood vessels that are very leaky around tumours, and get retained there and more into uptake at tumours. So rather than having to go and poke 20 different spots with cancer, there's a way to get these nanoparticles to hone in on and deliver them more specifically.
A research reported where they've actually used probably 3 techniques. They've used ablation of the tumour with cryotherapy or radiotherapy, an adjuvant to stimulate the immune response, then the immunotherapy, and they've actually used more than one. So it's really like a cocktail of trying to target all these blocks. And they have to identify it with imaging to find the right spot.
For sure. So that would work for a localized cancer. So if you had a localized lung tumor, that may work. But our challenge is oftentimes when the cancers are more widely spread, how does the work that Brendan and Mara described boost the whole immune system to go and attack all the tumor spots that are beyond what a local therapy can get to?
The discussion. The idea is actually to use the tumour tissue as an adjuvant, and then it's like a vaccine. The body can possibly then see it in other spots in the body.
And so so there are multiple strategies to develop vaccines of using the tumour and then incubating them with the immune system and then giving the immune system back. So you can either do it locally or generally. But what it speaks to is the importance of the immune system being a part of the cocktail. And the work that Brendan and Mara is doing is how can we optimize the body's own immune system as well as the therapies that we make in addition to attack the cancers. So you're absolutely right, it's a cocktail. And there's so many parts to this drug development process, from just the basic science to the pharmaceutical science and the new strategies to attack cancers in many angles.
The other consideration too is that every tumour is very different. So even within a cancer type. So if you just say gastric cancer, for example. So what may work, if you're talking about the inflammation or the immune response, in some patients, it may actually be good to spike the immune response because then you're going to obviously have anti-tumour immunity to attack the tumour through T cells. But in other situations, if you're actually spiking the immune response, you could be triggering inflammation. That inflammation could actually be providing the milieu to further drive the tumour. And again, this is why it really is important to understand the tumour and why a mantra that we're all very focused on is personalised or molecular targeted therapies. Because it certainly can be, yeah, whether you've got a cold. I like the cartoons Mara had of going from that immune desert to that lush landscape where you're going from a cold environment to a hot environment so you can actually have that immune response. So yes.
We'll move on. That young man just here.
Yes, there was a paper recently released internationally where there was half a dozen patients who responded to the CAR T process, where they took tumour and then specifically arranged the T cell process. Unfortunately, it cost about $600,000 a dose. I was wondering, do you think that's a realistic pathway to pursue?
Maybe I can answer this one. So there's a lot of work going on now about trying to generate more off-the-shelf cell therapies. So because of that exact reason we said, where it's a crazy amount of money and it also takes a very long time. So because they have to isolate a patient's immune cells, expand them in the lab, engineer them, and then infuse them back. So that's time it's wasted really with the patients waiting there, and depending on when they're diagnosed, it can actually be quite detrimental, that time period. So yeah, that's definitely something that people are focused on now, is trying to generate a set therapy where you can kind of have multiple different CAR T cell products that you can just grab and use on a patient straight away. But given, of course, we're talking about the immune system, you can get rejection of immune cells that aren't your own. So it's a barrier, but that's something that's very hotly being pursued at the moment. Yeah.
There's also been released information concerning a new immunopathology process, which I think was mitochondrial based. I'm not sure, but they've detected very short-acting proteins that are released separate to what you've provided here. Is that any comments about that?
Or if you're talking about the mitochondria, you're probably talking about metabolism. And that is a very hot field, especially immunometabolism, where now the immune system is influencing metabolism. There's a lot of research. It's still a very young field. So we're still in the early stages. We don't really understand how immunometabolism can impact on a lot of tumors. And one of the problems when you think about metabolism and the immune system, there are so many different types of immune cells that are present. And it's, you can try and classify metabolic processes through mitochondrial dysfunction, et cetera, and say T cell, but even within T cells, you have different subsets of T cells, and that's just talking about the adaptive immune system. But what about the innate immune system? So it's a very hot field of research, and there is still a lot of questions to be asked in that space.
Put up your hand so Gia and Anne can come and find you, but we're going to come to the gentleman here.
Good, thank you. About the innate immune system one up here. The results with your tumour model are terrific. With human tumours and with the advances in genomics, so many mutations that are drivers of the tumour, oncogenes or loss of tumour suppressors, are present. When there's a tumour with a clearly identifiable driver, an oncogene mutation, is there a role for the innate immune system? Are they still there? Are they still hanging around? Are they, instead of drivers, are they bystanders or secondary? Where do they fit?
That's an excellent question. And I'll actually give you a oncogene that's called KRAS that many people may be familiar with. So KRAS is present. It's a driver mutation in 90% of pancreatic cancers. It's probably about a driver of maybe 1/4 of all lung cancers. Now, KRAS mutation, it certainly is the initiating driver mutation in those cancers, but it's a single hit. Many of these cancers, you need a second hit. So the driver mutation alone isn't enough. It may start the process, get the ball rolling towards a cancer, but you still need additional hits. And this is where the immune system comes in. So it's certainly known for KRAS that when KRAS is mutated, the innate immune system is upregulated. Okay, so there's inflammation. There's a low-grade inflammation that is induced as a consequence of that mutation. So when KRAS is mutated, there's a whole range of pathways downstream of that, and that feeds in and starts to drive this low level of inflammation, and that inflammation supports and nurtures the growth of the tumor. Okay, so you need that inflammation present in the background of having that KRAS driver mutation. So definitely inflammation is not, I wouldn't say it's always essential for a tumor. There are probably situations where it may be more of a bystander, but there's certainly a lot of cancers and tumors, especially ones, as I said, driven by KRAS, where inflammation is playing a critical role in addition to the initial driver mutation to support the tumor growth.
47 people online, so I'm just going to have a shout out. Feel free to write in the chat box, and Catherine will let me know if you have a question. But a lady just here.
Are monoclonal antibodies used in the manufacture of the drugs?
Yes, absolutely. I think you've got some pretty good examples in immunotherapy you're referring to.
Yeah, so the standard, well, one of the standard treatments for lung cancer is these anti-PD-1, anti-PD-L1, and they are monoclonal antibodies. So that prevent that sort of interaction between the two proteins and shut off that inhibitory signal, and so allows the T cells to attack. So that's one of the things that patients get screened for is, do the tumors express PD-L1, and so then they're candidates for these monoclonal antibodies. Yep.
The beauty of the antibodies too, especially if your protein of interest that you think is driving the cancer, if that's expressed on the surface of the tumor, that's when antibodies are really good, because they can just attack on the cell surface and hopefully block the action of that protein.
Left.
Poor Gia, she's going to be tired by the end of tonight.
She's a young lady.
You haven't addressed how immunotherapy could help breast cancer. I had a daughter who passed away with breast cancer, and she was on tamoxifen for 10 years, and it seemed like everything was fine. But as soon as she stopped, it came back in a fury, and she was gone within 12 months.
Yeah, so breast cancer is similar to lung cancer, similar to a lot of the solid cancers, in that, yeah, very, very few patients have good responses to immunotherapy. So in most situations, for most doctors, it's probably the last point of call in terms of an option for treatment. And at that sort of stage, yeah, it becomes almost like a last resort of like, maybe it'll work for you, even though it doesn't work for a lot of people, but maybe you'll get lucky and it'll work for you. So that's the way that they've been treated at the moment in the clinic, which is a shame, because I think they've got a lot of potential, but as you can see, there's a lot of work to be done to get them to a point where they're not the last resort, and they're something that is used early on because it can manage and control the cancer. You get a lot less side effects. It's a much nicer treatment than something like chemo or radiation. So that is why we're sort of interested in trying to get it up to a point where it can be used in an earlier stage. So yeah, unfortunately, I think they all have similar issues to what I said about lung cancer. So they all have the ability to hide from the immune system. They all have the ability to express these ligands and to have almost no immune cells in their tumor, particularly triple negative breast cancer is particularly bad for immune cell numbers in the tumors. So all that really compounds into this really like terrible situation where it doesn't work. So yeah.
Is the availability of artificial intelligence tools transforming both the pace of your research and the sort of insights that you're able to derive simply by the capacity of those tools?
You want to answer on behalf of Elaine, who's not here?
So the answer to that is most definitely yes. The extreme varieties of the proteins and the structures, and artificial intelligence is helping us how to use the structural biology. And one of our medicinal chemists is here, Michael Roy, who's going to be using that. And then Lan is going to can possibly answer how we can use it to manage large data. So maybe Michael Roy can talk about how we're going to use it for developing drugs, and Lan can tell us how we're going to use it to analyze data. Oh, you start, Lan.
Okay, thank you. So my lab is also new to SAiGENCI, and we're very interested in developing new small molecule drugs that are known as targeted protein degraders. So they act by slightly differently to how many drugs work. Many drugs inhibit a protein. Degraders work by actually essentially tricking the cell into removing that protein entirely. But what we're doing together with some of the AI team at SAiGENCI, So Fui Lee's lab work on developing AI methods to, in this case, in the case of our collaborative work, is to improve the way that we can predict what sort of drug molecules we need to make to form really effective protein degraders. So in that case, we might be able to better. So normally you'll have to screen a lot of compounds to be able to develop a really effective compound drug. But using AI tools, we can hopefully streamline that process significantly so that as researchers in the lab, the number of compounds we need to make is significantly reduced, and then we can speed up development of new therapies. So I'll hand over to Lan.
Lan is in charge of computational. Oh gosh, I'm going to get it wrong and let you introduce yourself.
Hi, thank you for that question. So I think it's a very timely question. So my name is Lan Nguyen, and I'm heading up the computational system oncology program as part of SAiGENCI. And the program is all about utilizing computational approaches, including AI, bioinformatics, and mathematical modeling to analyze the vast amounts of data coming out from patients, and hopefully getting actionable insights that could help, understanding drug resistance and treatments, and coming up with a new way of treating these cancers. So just focus on the AI related question that you posed. Nowadays, it's become more and more straightforward to generate a huge amount of data from patients. And what we are going to do with this data, and AI becoming a more and more important tool to extract insight from this data. But there are challenges with that because, when you think about AI models like ChatGPT, image base model, there are millions of images that are available for training this model, right? And this model becomes so powerful because the data that used for training them is massive. Whereas although we have more and more data in the biomedical space, the amount of data that are available from the number of patients is still limited. And so there are challenges in terms of the availability of the data that could be used to train this AI model. So the answer is yes, AI is going to be as being useful and going to be very useful as the amount of data we get from patients become more and more accumulated, not only from each patient, but from increasing the number of patients we can profile as well. And so there are, consortiums that are at the international level where we can pool more data from different countries and forming a much more larger data set, and these are where the AI are going to become more powerful. So it depends on the data, which is critical. It's not it's not like you can solve everything now, but because it critically depends on the data. But I think in the future, in the next 5-10 years, when the technology become better and then more data, then this AI will become indispensable in not only cancer, but in any other diseases as well.
Thing to that, some people may have concerns about AI and machine learning and patient privacy and things like that. What I would just like to say is that whenever we do go down that path, we actually are very responsible with the data and how it's managed. And I'll just add to that and say, the Australian Institute for Machine Learning within the University of Adelaide, an amazingly well-recognised AI institute, is actually setting up something called the Centre for Responsible AI, so that you be aware that we are very serious about both investing in it, but doing it properly and safely. And we've got one last question online that Mara is going to ask, a very deep scientific question to...
Yeah, so the question was, why weren't we getting additive effects when we combine the dub deletion immunotherapy? Why are the two together doing the same thing? The two separately doing the same thing, we don't get an additional effect. And the other question was, if it's involved in the cell recycling, then wouldn't that then lead to a bit of a toxic drug if we were to use it? So to answer the first part, yes, actually, we were all surprised that there wasn't an additional effect. We would have thought that if we combine both together, that we'd get more clearance of the cancer than either agent alone. So we're still looking into the biology as to why that's the case and analysing those tumours in a bit more detail. That this data's all really relatively new. But really, at the end of the day, if that means that we can use either agent independently and still get the same response, then I think that's a real positive thing. So, or that we can use just a DUB inhibitor instead of immunotherapy as another alternative option for a patient, I think is good. And the answer to the last question, yes, it's true. So especially if a protein has multiple roles in the body, then you are opening yourself up to multiple side effects when you use a drug. But I guess that probably speaks to what the comment we heard earlier in the audience about targeted tumour delivery of said drugs. So that's probably something that we would have to look into if we do want to pursue this. Yeah.
Thank you very much, Brendan and Mara, for deep science. And I'll just say it's probably the first draft of history on some of the science that you saw today. And as I say, the Wright brothers didn't get their plane up on the first time, but they did eventually, and we're flying around the world. We're flying to space even. So you're seeing that this is an iterative process and it takes a while. Glenda's story tells us that we found a mutation in her tumor, something in the EGFR, I suspect, and it was very sensitive to a drug that was defined by a medicinal chemist. So she's an example of the advances we have made and will make, and more to come. But if the young man, Albert Einstein, had anything to say, he said many things wisely, one of which was, if we knew what we were doing, it wouldn't be called research. And with that, I would say, I'd like you to attend. Thank you for coming for this evening. It's been marvelous to get such a warm and generous reception from everyone in attendance, and your support is so greatly appreciated in any form. And we have research ambassadors rolling around with badges that says ask me questions, because I'm a research ambassador, red badges from all of SAiGENCI, the volunteers here tonight, and other scientists. Brendan and Mara are going to be here for any questions that you have if you're too shy to come up. And we have refreshments out the back there on the way out in the foyer. So thank you very much for your attendance, and it's very much appreciated from everyone.